Yes, what are other databases for the DNA sequences? GMBL, NCB, GenBank, yes, DDBJ, correct. Yeah. Okay, then for the literature databases? PubMed, correct, okay. And then for the protein data sequence databases? For the protein sequences? UniProt, right? Fine. Okay. So in this practical session, we'll show some demos and I'll give you some questions. How many of you accessed these databases? How many are familiar with these databases? Okay, anyway, I'll give you some questions and I will ask you to get the answers. If you are not able to get the answers, okay, we will show the demo and you can use it. Okay, fine. Are you able to see my screen? Yes. Okay. Okay. Fine. Okay. Mainly I discussed about the databases and features. I discussed about a tool today morning, right? So sequence to feature. So first I demo about the sequence databases and then followed by the tools. Okay, this is first part, the DNA sequences. Okay, I'll show the questions and then I'll ask my TAs to give the demonstration. The first question is, okay, dealing with the DDBJ and the GenBank and EMBL. Okay, so I have a few questions. You can go through these questions and look at these websites and you can get the answers. First question is about the how many Homo sapien sequences in DDBJ and compare the data with other databases. Second question is the GC content, because I discussed the GC content in the morning. There is total number of G plus total number of C divided by the total number of all the nucleotides. And the second part, literature databases. So here I have 6 questions so that you can be familiarized with the PubMed database. And the third part is the protein sequence databases. Okay, so mainly we use UniProt, right? Although PR is the first developed database along with the SwissProt, and finally the unified to UniProt, right? So I have 6 questions, and we will get the answers for all the 6 questions, so that you will be familiar with the UniProt database. And I discussed about the tool, sequence feature tool, right? Very briefly I discussed due to the limitation of time. So now we have more time now, and I gave a lot of questions because I have 3 sequences and 5 questions for the protein based. Okay, we'll explain all the details, right? And then you can get the results and you will be familiar with the all these tools. Okay, fine. So let's see, and then again I'll go with the same sequence feature, the DNA based features. Okay, here I get 3 sequences. I specifically mentioned 3 sequences because these sequences are different. So can you see these last 2 sequences and tell me your comments? Do you find anything different in the last 2 sequences? What's the difference among the 3 sequences? Am I audible?
Yes, sir.
Yes. You see these 3 sequences, can you find any differences? Correct, right? The last one, second one is completely AT, third one is only GC, and the first one is a combination of all the nucleotides. Because depending upon the nucleotide sequences, your properties will change. Okay, that is the reason I deliberately gave 3 sequences. You get these parameters, and from these parameters, right, you can see, okay, how the values changes on the different sequences, and whether these parameters are useful to discriminate different types of sequences. That is the reason why I put all 3 sequences. Okay. Can I ask, Mehta is here?
Yes, sir.
Yes, Mehta and Kulanda are here. They are my TS, and she will explain, right? So all the details. And first, you try to do it. If you are not able to do, then she will tell you how to do it. Okay, so then you can familiarize with all these databases. Okay, yeah. Meda, yeah, Karyan, I will.
Yeah, thank you, sir.
Yeah, okay. Stop the screen. Okay.
Yeah, so good afternoon, everyone. So this is a demo session, and we'll try to explore several databases which were previously discussed in the lecture session. And so, as sir already mentioned, that there are several databases like DDBJ, GenBank, and EMBL, which are mainly for DNA sequences, and PubMed for literature, UniProt for protein sequences, and Seek2Feature is a tool which is used to derive sequence-based features for DNA as well as protein sequences. So I'll try to share all the possible information, whatever you will be needing for your demo, and I would like to ask you to do all those things in your browsers, and if you have any doubts, you can ask in the chat box. So I would start. So I'll share you the links first of all. So moving to the first question, we have this question where we have asked for the number of sequences which are available for Homo sapiens in case of DDBJ, GenBank, and EMBL. So now I'll share you the link for GenBank in chat box. If you can you can like directly explore it in Google. And if you don't find, I'm sharing you the link for DDBJ here in the chat box. You can copy and paste this in the browser, and you will redirect it to the DDBJ website. So, and if anyone has any doubt, they can ask in the chat box, or they can unmute themselves and ask the questions. So, moving ahead to the next slide. Next slide, I'll show you how DDBJ looks like. So, if I'll give you some time to explore it, and if anyone is not able to do that, they can ask the question in the chat box. So this is a DDBJ homepage where you can see different options such as search and analysis, submissions, download, supercomputer, all those things. And we are mainly focusing on Homo sapiens. So we'll go to the search option here, and we'll once you click to the search option, you will again be redirected to several resources present in DDBJ. So such as get entry, ARSA, DDBJ group cloud, and other stuff will be there. So this ARSA is a sequence retrieval system. So ARSA has a full form which is all-round retrieval of sequences and annotations. So it has several DNA sequences. Now you go to the search option and type your keyword Homo sapiens, and once you click to the search button, you will find several sequences. So the first blue colored keyword is your accession number, and you will find a list of sequences which you can download in different formats. So is there anyone who is having issues with DDBJ? Please let me chat box.
Are you able to access the website, participants? Okay, good.
Yeah, so once you are done with the exploring, you let me know how many sequence entries you found there in the DDBJ. Someone mention in the chat box so that I'll get to know that, okay, you are able to find it. Okay. So yeah, someone wrote 60 entries. So 60 entries will be initial 60 entries. There will be a lot more other entries. So you can download, like it is displaying 60 and first 60 entries right now. But you can see there will be other entries. So there are around 32 lakh entries which are reported in DDVJ. Yeah, correct.
Right, yeah, right, yeah.
So I think people are able to access. I can move ahead to GenBank now. So moving ahead to GenBank, again I'll share you the link for GenBank here in the chat box. And if you can directly explore in Google, or I'm sending you here in the box. So this is the link for GenBank. And once you click the link, you will be redirected to this page. You can select the database as nucleotide, and you can search for your keyword Homo sapiens here. So once you select Homo sapiens, it will, like, it is always good to give the organism's taxation ID. So it will specify a specific number of Homo sapiens sequences. Otherwise, sometimes keyword takes other entries which are related to Homo sapiens. So if you give the taxation ID, which is for human, it is 9606. So if you give the taxation ID there, you will be getting a fixed number of sequences which are reported there in GenBank. So once someone is done, please mention in the box if you are able to search the number of sequences which are available in GenBank for human being. Has anyone done yet? If anyone is having difficulty, they can unmute and they can ask the question if there is any.
Who got these numbers?
Correct. So it is close. But yeah, it's correct only. So is there anyone who is having difficulty here in this GenBank? Or we will proceed to the next database. We'll check for EMBL now. So EMBL is mainly managed by EBI, and again it is also a DNA sequence database. So I'll share you the link for EMBL.
Ma'am, can you please explain how to find out the sequence in GenBank? Actually,
Yeah.
I have typed that Homo sapiens.
Okay.
Yeah, please explain how to find out the.
Yeah. So you search for the keyword Homo sapiens there, right?
Yeah.
You change the database also as nucleotide database, as mentioned in the screen right now.
Yes, right.
Yeah. So once you are done, you just click the search button.
Yes.
Yeah. So after clicking the search button, in right side, you will get the information about like based on taxonomy, organisms based on taxonomy. If you check right side in your screen.
Okay, all right.
Yeah, so once you click that, you will get the specific number of Homo sapiens sequences which belong to Homo sapiens. Otherwise, sometimes there will be other related entries which will be reported.
Okay, can you repeat again that something you have written POR?
Yeah, so that is taxonomy ID. I'll explain you later on how you go to the advanced search and how you can select for all these things. So right now, once you did this, you would be getting a query like I am getting on top, right? You can see this Homo sapiens taxonomy ID 9606. So similarly, in your page, you will be finding a query like this. Just look at it and tell me if you are finding the similar kind of thing or not.
Okay. Yeah. All right. I got it.
Yeah.
Also, could you click one of the results and then go show the contents page? What are the to show the contents of this database? Because morning I did not have much time to show the contents.
Okay. So, Kulani, can you share the screen there and show them? I'll share one link from here. You can see it in chat box. I'll just give me some time. Meantime, if anyone is still doing that, you just do. I'll share you the content of any file there.
Okay.
Click on any one of the results and then just go inside. Then it will show all the contents. Anyone is fine.
Yeah, one second.
I showed in the morning about the contents of these databases.
Yes.
And due to the limitation of time, I couldn't explain the details.
Yes, 1 second.
Anyone is fine, either the DDPG or GenBank or EB.
I'm opening a data file for GenBank here, and I'll share you the link. Everyone can open the same link so that it will be easier for everyone to understand. So I have shared one link here for one GenBank entry. So this yeah.
Mega, are you able to see my screen?
Yeah, I am able to see your screen.
Yeah, this is the contents of the database. These are the information which are available in the database. You can see the definition and the source is Homo sapiens and the reference where they published the sequence, right? So you can see all the details in the contents. Can you go down? Yeah, so here they give some comment and the features. Yeah, okay, just go back a little bit. Yeah, go down. Yeah. Okay, fine. Yeah. So here you can see the full sequence. But GenBank, they did not give the number of ATCG, but in other databases you can see the number of ATCG and all. Okay. So are you the participants are you able to get these pages and then check the contents?
Yes, sir.
Okay, fine. Okay, fine, Meta. So maybe you can go back to these slides.
Yeah. So someone mentioned, Rashmi, are you able to do this or still you have an issue?
Yeah, ma'am. I got it.
Okay.
Fine, yeah. Okay. Then when you find time, you can go through the details of all these three databases and you will get all the contents and we have various search options, advanced search options, and you can use that to familiarize with the databases. Yeah.
So next we'll be moving ahead to EMBL. So EMBL, once you click this link, you will be redirected to this page and you can search for you can go to the search option here. And once you go to the search option, search for Homo sapiens. So on top, you can see the search button where you can write the keyword Homo sapiens and you can search for the sequences which are there reported in EMBL. And someone please mention in the box like how many sequences did you get?
Good.
So is it okay for everyone or is someone having a difficulty in this database? So you would have noticed that there are differences in the number of depositions in different databases.
Yeah.
Okay, so we'll
Yeah.
move ahead to the second question, where we will try to get the GC content for a specific sequence. So here we have given the accession number of one particular sequence. I will share this accession number to you, and
Yeah. Meda, just stop. Okay, so now Meda gave the question that you have to get the GT content of this one. Could you find and give the answer? If you are not able to get the answer, I will ask her to show the demo. Participants try to use this accession number and then try to get the GC content. First, you write how many G's and how many C's, and then you can calculate the GC content.
Yeah, so for you, I'll give you one hint. Like you already checked that GenBank doesn't have individual ATCG count or maybe GC content, but you can check in DDBJ. You will get the number of bases available in one particular sequence. So I shared the DDBJ link before. So you can use that and go to the search option, look for the accession number, and get the file in which you will be having the information of information as we showed you before for GenBank.
How many G and how many C? Is this number correct?
Yes, sir.
Okay. Good. Okay, for others, maybe Meda will show the demo.
So how many CR72, GR129, and TR100 and AR131? So those who found, it is okay. Those who couldn't get, so we have represented here in the screen. You can see, once you go to the DDBJ, look for this entry, and you will get the details as you were getting before. Once you click to this accession number, you will get the file as shown in the screen. So if someone is not able to do, they can tell. Otherwise, we'll proceed to the next question. So next question is about getting the papers from PubMed. So here we have given one search which is about description of beta barrel membrane proteins. So you already know that you get the information of all literature from PubMed. So you can open PubMed in your browser, and I'll share you the keyword search which you have to search there in PubMed. And if you need, I'll share you the details of PubMed. I'll share you the link. So here is the link for PubMed and the keyword search which you have to do. someone who is maybe from different background than biology can tell if they are able to find. They can unmute themselves and they can mention if they are able to follow or not, so that I'll get to know, okay, it's working fine. Okay, so are you able to go to the link which can you just follow the link I shared you? Can you just click to the link and go to the PubMed page?
Yes, ma'am. 47 results.
Sorry?
Yes, I
Yeah,
got
you
the
got
page
40.
which you have. Yes, 47
Okay,
results.
okay. And yeah, so okay, okay. So I think everyone is able to do that. So now next question is also from PubMed. So here, like you have to find papers which are published by, like let's say example we have given for Nasinovar. So if you check for this fellow, you can look how many papers he has published and which are there in the PubMed. So okay, some people are in the previous question right now, but I think everyone is able to do that. Yeah, so again, if anyone is having issues in this question, yeah. So yeah, here if okay, you would have tried it directly. If I think we'll go for advanced section in next. Is there anyone who is having difficulty in this one, or we'll proceed to the next one? Okay, okay. So next question. Next question is again from PubMed, where we'll be checking for this specific entry. So if you can see here, we have mentioned about one specific paper which has been published in Cell in 2008, and date is also mentioned, which is December 26. Hello, am I audible?
Yeah, yes, madam.
Okay. So I'll share you this detail, and I'll share you the detail of the paper, and I would like to ask you if you are able to get the information about this. So in this question, we have to check for the related entries, how many related articles you are able to find for this particular paper. So if you search for this paper in PubMed, in right side you will get similar entries about this paper. So it will suggest you some similar articles with this paper. So here you can see that you can see the details of the paper. Once you go to the similar articles, you will find the list of the papers. So we'll wait here for 1 minute so that everyone can do. Yes. So this is one paper. So it is already mentioned that this paper is published in 2008 in Cell. Yeah. Is there anyone who is having difficulty? Then please let us know. Yeah, so we have asked for related articles. Anupam, you can check like how many related entries are there. This paper is single paper. In the right side, you will find a link for similar articles. Yeah, I think everyone is following, so we can go to the next question. Next is again from PubMed, where we'll try to find the papers which are published in Nature for year 2020. So I don't think so I need to give anything here in this question. You can directly find. Now we'll try to use advanced search for this question. So you can see when you go to the PubMed homepage, you will find advanced search option there. Kulandi, just show them where that advanced thing is there if they are not able to check. I think in previous slide, we mentioned that there is an advanced search option. Go to the previous slide once. One more. Once. Yeah. So once you go to the PubMed website, you will find this advanced search just below this search option. Just click to that and now move to the next one. Just click to that, you will find a search query there, like what all options you can look for. So you can add different terms to the query box. So in the drop down where in the screen you can see that date publication is mentioned. If you will click to the drop down, you can find journal there. So once you go to the journal, you can write the name of the journal in the right side. So Nature is name of a journal where different paper publishes. Okay, people from different background, I want to know from the people who are not from biology. Okay, so sir, I think you checked for all the papers which are there in Nature. So we mentioned that we are looking for papers which are from year 2020. So if you give the specific date of publication, you can go to, then you will be getting a query box where you can mention the date and then you can search. You will find limited number of papers which are published in 2020. So Aisha, are you able to do that?
Yes, ma'am, but I don't find any relevant papers of my research. Like, what means that's why I'm not able to link all those things.
You are from biomed. You will definitely find papers in PubMed where you can
They are purely
which will be
medicine
related.
oriented, ma'am. They are medicine oriented. We want engineering background, ma'am. Processing
Even for
like.
engineering background, you will find papers in PubMed.
Okay, ma'am, I'll try.
Yeah. So later on, yeah.
Yeah, ma'am, I'm the coordinator speaking. I am from mathematics background.
Okay.
As madam previously said, maybe engineering and mathematics related some papers, like some statistics being used to deliver some product or to discover something like that. How to find those papers? Because if such papers are there, then we can connect it with our mathematics.
Okay, so you will be knowing the specific journals where your papers are being published, right? So this
no,
is one.
No, ma'am. No, we don't know, ma'am. That's what is the difficulty.
We need your support to find out our, our area related papers. maybe
Oh.
from mechanical engineering or biomechanics or biomedical image processing, something like that. So if you do one or two, searches for us, that will be really great help.
Okay, so would you like to suggest us some keyword for which
Ma'am, for example, modeling COVID.
Okay, correct. So we'll try to show you that.
Disease spread.
Okay, fine. So meantime, other people will be doing this thing.
Ma'am, few more things also, like machine learning is coming up in this, no? So you may be knowing. So those papers also, it's fine. So actually, what we need is just a small connection between mathematics and biology, so that we can hold on to it and we can get into it. Something like that.
Correct.
Yeah,
so we'll try to show you the same, like similar kind of thing.
Okay, I'll answer the question. Okay, actually the PubMed is mainly for the biological sciences. You can get almost all the journals which publish biological sciences papers. You can get in PubMed. Okay.
Okay, sir. Okay.
If any mathematical models or biomedical engineering or any of the papers, okay, if they publish in these type of journals, you can get from PubMed. There are a lot of other databases, for example, Scopus, or you got the Google Scholar, or you got science citations. Okay, these are the other data resources which covers everything. For example, you go to Scopus, they have specific options for engineering, and they cover almost all the fields.
Yes, sir.
Yeah. If you use PubMed, you may miss several articles, mainly the engineering and mathematics side. For that, I suggest to use the Scopus. Maybe, Kulambi, can you go to the Scopus site?
Yes, sir.
Scopus is free. Anyway, Scopus is free, and the Google Scholar is also free. And only this science citation index, that is a paid one. So you can use Scopus, you will get all the information there. Okay, Meda, you can use the PubMed to related with the mathematical modeling and the COVID-19. Then you can get some papers here. But the same thing, if you search the Scopus, you'll get more papers.
Yes, sir.
Okay.
Yeah.
Okay, sir. Okay, thank you, sir.
Yeah.
Yeah, so here it's just a representation.
Yeah, we go to Scopus. Okay, this is a Scopus site. Okay, here if you check the list, you can see it covers all the engineering, and science.
Pulini, maybe write COVID, machine learning, or COVID, mathematical modeling, something like that.
Yes. I search for machine learning.
Okay, correct.
Okay, you can see, so maybe you can IEEE journals also, but the PubMed doesn't cover this type of journals.
Okay, sir. I got it.
Yeah. So go to the list. What are you saying for list? Or there are some disciplines also there. You can check that. Yeah. Subject you can see computer science, engineering, mathematics, physics. So there are various aspects you can see in the left hand side in the subject area. Yeah. So now you can see many biomedical engineering. Yeah, correct.
Sir, in this field, is there research going on in your lab, sir?
Yeah, actually, we are working on machine learning. We are working on mathematical modeling. We are working on COVID-19. Yeah,
sir. This would be very interesting for participants. So, as I earlier pointed out, we are having diverse participants. So participants can note this, and we can take a cue from sir. Yeah, he has a published paper here too, so you can just.
That is correct. Yeah.
So this would be very, very informative for participants.
Yeah, if you see my papers here, we have 281 papers.
Okay. Okay, sir.
If you click on that, you will get all my papers regarding the my work. Yeah, Scopus is actually, there are two different subscription for Scopus. First, basic one is free, and the advanced one is you have to pay it. But if you review any of the articles for Elsevier, then they will give you 1 month free access to Scopus. But the basic search and everything is free for everyone.
Yeah. Okay, sir.
Yeah.
Probably we can download this paper and we can go through it and approach professor afterwards.
Yeah. These are all
Yeah, sir. Yeah, sir.
Yeah.
Thank you, sir. Thank you, sir.
Okay. Yeah, this is the one. Or you can use the Google Scholar. Google Scholar also you can get all the information on all the fields. I think you might use Google. Google, I think you are familiar with that. So that if you go to Google Scholar and if you click my name, Kulasamy. Yeah. If you click it and then any search word you can put it, then you can get all the papers related to that. Okay, you can get the citations also. My papers are cited maybe in 10,885 times. Yeah, this will give you that information, each papers and all details. Any mechanical engineering or the biomedical, any papers you can get from Google Scholar. Okay. Yeah, whether you can proceed to the next one. We don't have much time. We have only 45 minutes left.
Yeah, okay, sir.
Yeah. Thanks for the questions. It's good. Please feel free to ask any questions. We are happy to answer.
Thank you, sir. Thank you, sir.
Yeah. So this was the search for Nature publication in 2020. Now we'll move ahead to the next kind of databases. So in previous session, you already discussed about protein sequence databases. So you studied about UniProt, right? So now I'll give you the link for UniProt and we will discuss some of the problems which we can explore. So for the first question, we have asked for the amino acid sequence of human mitochondrial beta barrel membrane protein, VDAC, and its functions. So UniProt helps us to give all those details. So I'll share you the link and one can explore in Google. This is the link for UniProt. Once you go to the UniProt, you can search for VDAC protein. So if you write VDAC and you search you click to the search button, you will get several information in the left side. There will be a filter in the left side, and this will be the details you will get there. So it's a voltage dependent enhanced selective channel protein. It's a membrane protein, and its function is also mentioned there in the next box. So UniProt will be having information about sequence, its function for most of the proteins, about their structures if it is available, their interactions, their subcellular locations, where they're located in the cell, and several other features will be there reported there in UniProt. So in this question, we have asked you to get the information about functions, sequence, and its how many transmembrane segments this particular protein has. So once you go to the sequence section, you will get the sequence of this protein. So sequence section is in the left side. In the filters, you can get the sequence option. So it has a sequence of length 283 residues, and it has several transmembrane segments. So someone can like immediately mention how many transmembrane segments it has.
How many transmembrane segments in this protein? Can you even count? Okay,
Yeah,
good.
correct. So it has 19 transmembrane segments. So you can explore several other features also in UniProt. So once you go to the UniProt site, you can check for different options there in the site.
This protein is very unique because usually the bacterial beta barrel membrane proteins contain only even number of beta strands, 8, 12, 16, these are 22, these are the common numbers. But here it is very unique because 19 is odd number because it is the mitochondrial. And why it is odd? Because the last beta strand and the first beta strand forms hydrogen bonds to form a barrel. That is the main aspect of this protein. That is the reason why I gave this for the understand the functions. Okay, carry on. Yeah.
Yeah. So we'll move ahead to the next one, where we will try to get the sequences which are having 50% sequence identity. So talking about sequence identity, it is like those sequences which are more than or like up to 50% identical with respect to, like, if we are checking for two sequences, they will be identical. So same residues will be there up to 50%. So for this case, you can check for the keyword transcription factors there. So you search in the search bar for transcription factors, and then you can look for reviewed entries, which is mentioned in the left side. Reviewed entries will be there. Once you click to the reviewed entries, you can go to the UniRef. So it's like UniProt reference proteome. So there is a section of UniRef in the left side. If you go to the bottom left, you will find UniRef there. If someone found UniRef, just mention in the box. Yeah. So, and if someone is not able to find, then also please mention in the box so that I can just tell once again. Okay, I think people are able to find. So you have to check for 50% sequence identity. If you click to the 50%, you will get some information. So I'll explain that information to you. Just once you are able to find that, let me know. So once you start checking for 50% sequence identity, you can see your query on the top. So it gives in query that and identity 0.5. And in the left side, the highlighted box, you can see how many number of clusters are formed after doing 50% identity check. So someone will mention in the box how many clusters you found. Okay, so you found 4515. That might be because you would have checked for all entries rather than going for reviewed entries. But it is good to explore. You can check in different ways, and the number can vary here based on your query. So query is on the top box, highlighted box, where you do the search. So here it is mentioned in the question, list the count of sequences and count of clusters. So can someone tell me how many sequences will be there? The count of sequences, can someone mention here? You can see a column named as size. So every cluster has their own like different size. Let's say first entry here is 6. So this particular cluster has 6 sequences. Next cluster has 15 sequences, and so on and so forth. So if you will check in your list of like your table, you will find different numbers. Once you add all those, this will be the total number of sequences. And basically this number of sequences will not change. The count of clusters only will vary.
Ma'am, actually, it will be better if you'll show that particular website, visit that website, and it will you'll show that, because we are non-biological background. We are from non-biological background, so it is we are finding difficulties while
Okay.
finding your following your questions.
Okay. So
Instead of presenting the slides, it will be better if you'll visit that site and show the things.
okay, fine.
Kulanthi, can you
So-
go to the site and then just show that?
Yes, sir.
Make the next 2 questions you skip because we don't have time.
Okay, sir.
Similar to next 3 questions you skip, then go to the statistics next.
Okay.
Yeah.
Whether are you able to see my screen?
Yeah, you are presenting. It will come in. Yeah. So
you can see the
correct.
transcription.
Kulanthi, put it in quotes so that they will get same number. So if you don't put it in quotes, it will consider those two as different keywords, separate keywords.
Yeah, fine. It's
So if-
okay.
Yeah. Now you search.
Then I have to select only the reviewed sequences.
Yeah.
Then I have to get the only 50% of sequence identity sequences. So just you have to click the here, the 50%.
So are you able to do it now?
Yes.
Okay.
Yes, sir.
Okay, good. Yeah, it's good. Yeah, good that you mentioned. It's easy. If you are also, it is easy to show. Okay, so you got the data now.
Yeah.
Hey, what
So
do you-
here you can see this third column where you can see the size. So different clusters have different sizes. And this total will be giving you the total number of sequences which are there in with 50% identity. Like it will be total set of sequences actually.
Because why do you need the 50%? Because when you do a large scale analysis, for example, if you have thousands of data together, and if you use the same data many times, you will introduce a bias because there is an influence of that particular sequence, right? So that is the reason we need to have the sequences which are totally independent of each other. You cannot completely eliminate the 0%, right? So we can have some cutoff. 100%, 90% is very high. So 30 to 50%, depending upon your data size, you can use it. That is the reason why we go for 50% sequence identity.
Is it clear for everyone now?
You got the data? 50%? Okay, good.
Okay, so we'll move ahead to the next one, where we will try to look for the statistics of the data which is available in UniProt. So if you go to UniProt homepage, so Kulanthi, just skip 2-3 slides.
Yeah, just
Directly go to the statistics.
question number 6.
Yeah, so you okay.
This is a question number 6.
Yeah, so here you can see you have to check for the statistics which is reported there in UniProt. So they have put all the information whatever there is UniProt in form of a statistics page, and you can see the summary of the data reported there. So if you scroll down in your home page of UniProt, I think Kulinde, you have to show or they can do. I'm not sure.
Let them do. Just go to this page and go to statistics and give the answers for all the 3 questions. If you're not able to get it, then we will show it. So, Kulinde, go back to the previous page. Okay, yeah, these are the 3 questions I asked. Go to Uniprot. Can you type the Uniprot URL in the chat box?
Okay.
Yeah. Okay, participants, now go to that website and go to the statistics and then get the results for the 3 questions.
So this is the URL for Uniprot. You can directly search for keyword statistics in the home page. If you're not able to find it, then go to the stats page and check for the different information reported there. And try to do these 3 questions. Has anyone reached to statistics page?
Yes, ma'am.
Okay, go on.
No.
Or
Whether
But
directly
ma'am,
to the statistics website.
yes, statistics website I got, but the thing is distribution
I'll
of
give the name.
sequence length.
Yeah, just try to explore the same page. You will get the information. Otherwise, I'll show you.
Okay, ma'am.
So here I'll share you the link of statistics page directly. You can go to, yes. So once you can check for this old style, it will be easy to understand. It will be in a pictorial representation. Justin mentioned in the chat box this Uniprot KB or slash Swissprot statistics old style. Go to that link and you will get the details. Yeah, so Rashmi mentioned here the shortest sequences of 2 amino acid residues, whereas longest sequences of approximately 35,000 amino acid residues. If anyone else found that.
Yes, ma'am.
Okay. So Rashmi, you have already found this question where you got the information about shortest and longest. So first question is about distribution of sequence length. So are you able to see the distribution of sequence length?
Yes, ma'am. Yeah, I am able to.
Yeah, so good. So can you see such kind of distribution there in the page?
Yes, ma'am.
So can you infer something from this page? So this is common for all. I will not get the reason of not having the same background. This is just a graph where they have represented the number of sequences, and in x-axis they have mentioned sequence length. So can you infer something from this graph? Yeah, someone mentioned that most sequences are of less than 500 base pairs, correct? Is there anyone who is having difficulty doing this?
Yeah, ma'am. Yeah, what that y-axis represent here, ma'am?
Running,
Sorry?
madam, actually running like a train. So go slowly, step by step.
Okay.
All if you will open all the sides, we cannot understand anything. Only 2 or 5%. Only 2 or 5% are answering. All are not able to answer.
Okay,
What
that's
is
why I
going on
asked you to mention.
actual on which
Okay,
way, what is
fine.
going
Okay, we'll try to go slow.
Yeah, ma'am, what that y-axis of that graph representing?
So y-axis represents the number of sequences. How many sequences are there?
Okay, all right.
So this is a distribution of the sequences which are reported there in UniProt, and it talks about the length of the sequences and the number of sequences reported. Yeah. Pulani, move to the question slide. Yeah, so some of you told me about the distribution of sequence length. Some are discussing about the longest sequence. Now, third is about amino acid composition. So, can like someone looked for amino acid composition also there in the page? You can see the amino acid distribution also in the same page. If you scroll it down, at the bottom of the page, you will find the amino acid distribution statistics. So those who are able to see amino acid distribution statistics can tell about the composition of amino acid residues which are which are distributed there in the database. So what is the composition do you find? Which kind of amino acids are higher or which are less there in the database? Okay, someone wrote aliphatic are more in number. So, okay. And so which one is the least reported amino acid residue? Okay, cysteine. Someone wrote sulfur base. So cysteine and threonine. Cysteine and cysteine and methionine. Where is methionine? Methionine. Yeah, methionine is also less. And tryptophan is least. Yeah, so is everyone able to follow this page? Okay. Those who are not able to follow, they can mention that, okay, they are not able to follow. So if everyone is fine, we will proceed to the next tool, which we discussed in the lecture session, that is seek2 feature. Okay, lysine is the highest. So, no, lysine is not the highest. So if you see properly, leucine is the highest. Yeah, okay. Yeah, all right. So this is simple representation of the data which is available in UniProt for amino acid composition. So Meantime, I'll share the link for seek2 feature. So this database, this tool has been developed in our lab, and it gives the information. It gives mainly the features which are like which we can get for protein sequence or DNA sequence, and which can be used in machine learning. So someone was asking here like how to do things in machine learning or all those things. So to resolve such kind of issues, people can get the related features for protein and DNA sequences from using this tool. So I have shared the link for this tool. People can click this link and just um, go through with the page for 1 minute, and we can proceed for the questions given for this. So first of all, we'll go for protein based features. So we have given 3 problems here. We'll discuss one here and we'll give rest of the 2 for your assignment purpose. You can do that later on. So I'll share you the sequence, and you can see the details first before using the sequence there. So just read the information written there thoroughly and you will get to know how to perform this. So if everyone is able to open the link, I'll tell you what to do. So those who are not able to do, I'll suppose they'll inform me that okay, they are not able to do. So I shared the link for the tool as well as one of the example sequence which you can use here in the seek2 feature tool. If everyone has opened this seek2 feature tool, they can go to compute section. And in compute, we are going to do for protein sequence. The sequence which I mentioned here is a protein sequence. So you can click to protein sequence. There are 2 options, protein and DNA. Go for protein. You can see the details mentioned here in this page, where they have mentioned about amino acid properties, substitution matrices, pairwise properties, and contact potentials. So we will calculate all those properties from this tool. So it is easy for people, those who cannot do, maybe those who are not very familiar with programming, they can use this tool. So after clicking the link, you can go to compute section. In compute, there are 2 options, protein and DNA, as shown in the screen. If you go to the protein, you can go to the, once you go to the protein section, you can calculate average property. So there are 3 options, detail, change upon mutation, and average property. So you can go to average property. It's highlighted in the second image here. You can see on the screen. If you click to the average property, you will get a box where it is mentioned that to compute average property values, provide the input sequence in FASTA format. So can anyone specify what is FASTA format? Is there anything specific about FASTA format? Someone can speak up. They can unmute themselves and tell. So FASTA sequence will have a greater than symbol. So before writing the sequence, you need to give it in a format where it will be greater than symbol. Yes, correct. So greater than symbol, in next line you will get the you will keep the sequence. So it is shown in the screen. First line greater than sequence, next line protein sequence. Is anyone not able to follow? So once you give the sequence, you can select for this amino acid properties and submit button. You just click to submit. Okay. So Sushma, where are you right now? Like up to which stage you did this thing? Did you check for the link? You can unmute rather than. Okay, so you clicked on average. Correct. Then you have to give the sequence. So I mentioned the sequence above, right? Are you able to see the sequence? Yeah. First give the greater than symbol and next line give the sequence. After then you can check to the button this amino acid properties. You can check this and then you can submit this. You can click to the submit button. Are you able to do now, Sushma? Okay. So once you are done, so someone would have got the results by now. Okay. So did it give this error to you? Previous Professor Charudat, you have some issues, I guess. Did it give short sequence length error? Okay, so did you give this in the same format? First line greater than symbol, next line sequence? Okay. Yeah, I'm also checking if it is giving any error, I'll let you know. But it gives the result. So if you give it in the proper format, greater than in first line, second line sequence. You can see in the screen, first line greater than symbol, next line sequence. Just go to check the amino acid properties, go to submit button, you will get the results as shown in the next figure. So Gurpreet, what is 0.0? You got output as 0.0 or can you unmute yourself and tell, because what is this 0.0?
Its value.
So when I am doing this, I am finding 4 columns. First column is about code, second column is about properties, third is about actual, and fourth is about normalized value. So
it's saying average property value for whole protein sequence. And value for it is 0.0.
Kulandi, can you just show them in a demo form? You just click this link. It's working fine. I think you will be able to do.
Yeah, yes.
Yeah. So everyone, please see, he's giving a demo like how he'll be searching. So this is seek to feature page. He is going to proteins. After going to protein, he'll go for average property. Here he will give the sequence, first greater than symbol and second sequence. Now he'll check for amino acid properties, red colored sentence. Then submit. Once you submit, you will get the results as shown in the screen. Gurpreet, can you follow now?
Yes, ma'am.
So did you get the result?
Yes, ma'am. I'm trying it. Just let you know.
Okay, fine. So yeah, meantime she'll try. We'll proceed to the next one because we'll be having lack of time. We have 2-3 more questions. So if anyone has doubt, they can ask later on also. So moving to the next question, we'll check for some mutations. So here we have given some mutations here. You can get the property values for these mutations. So again, we'll check for first option, this D3A, D3E. So these are two mutations where aspartate has been mutated to alanine and glutamate. So I'll write it here. You can use it while doing. So I'll tell you what to do after this. So these are the two mutations which we will try to look for. So this will be followed for the first sequence what we gave to you. Right now I have already given this sequence to you. So you can go to the Seek2 feature home page, main page. You can go to compute protein. After proteins, now you go to change upon mutation. Among 3 buttons, go for the central button and click that. You will get 2 options now, one for sequence and another for mutation with position. So in first box, you have to write the same sequence what you wrote for previous question. Greater than symbol in first line, sequence in the next line. And in the next box, you have to write mutations with position. So if at all anyone is not able to do, please let me know. And then click for amino acid property same as before and then submit. Yeah, anyone is having issues, they can tell me. We'll wait here for 1 more minute and then we'll go ahead for the next one. Anyone is not able to follow? Or anyone got the results? Okay, Sushma got the results. Anyone else? So, Sushma, what all information did you get in the result section? Can you please tell me? Anyone can unmute and tell what information did you get?
Hello. Yeah, ma'am. We got some code properties, then D3A and D3E. Those 4 columns we got.
Okay. So can you understand? Okay. Yeah, Sushma, tell me.
Compressibility, thermodynamic transfer, hydrophobicity, surrounding hydrophobicity, polarity, isoelectric point, molecular weight.
Okay. So many properties you got, right?
Yes.
So these properties are for these 2 mutations. 2 columns are for mutations. So are you able to understand like what information you are getting from the tool? So these are different properties which tells the behavior of amino acid. And this tool is helping you to get the property values for these mutations. Hello? Hello?
Yes, ma'am.
Yeah, okay.
Yes, ma'am. Got it.
Okay, so I think everyone is able to follow. We'll move ahead to the next section of the Seek2 feature, which is DNA based. So in DNA based, again, we'll try to do things for DNA sequences. So here 3 sequences are given. We'll try for 1. and rest too you can do it later on. I'll give you the sequences in the end. So I'll give you the first sequence here, which is a combination of all the nucleotides. And you can go to the compute section in the same tool. Now, till now you were doing in protein. Now you have to do it in DNA because this is a DNA sequence. So once you click to the DNA, you will get the page where you can give your sequence, nucleotide sequence in FASTA format. So again, format goes like this. First will be greater than symbol and next will be sequence. So this will be the format to upload the information. This is a nucleotide sequence. So make sure while selecting, you will go for DNA sequence. After giving the sequence, you can select for physicochemical properties and submit. And once you submit, you will get the property values. If someone gets, please mention in the box. Yeah, so someone got properties, scale unit and average value, correct. So here you are getting property values for different nucleotides, like for different sequences. And now we'll check for the substitutions in case of nucleotides. So when some particular, let's say here example, this T has been substituted to A, T4C, G6C and G6A. So we'll try to get properties for these mutations. So again, sequence remains first one, and I'll give you the list of mutations. So you can see in fourth position, T is there in the sequence. So we are getting two substitutions which are in fourth position, and this T has been substituted to A or C. So what happens when these are substituting from T to A or C? Or in sixth position, G is there in the sequence. So what happens when this sixth position G it changes from G to C or A? So again, you go to the compute section, go to DNA. After clicking that, again you will get the same format as you were getting in proteins case. So you have to give protein nucleotide sequence in FASTA format, and you have to give substitutions. Once you do that, you can calculate physicochemical properties. You can check that box and you can submit. And so someone got the results. So yeah, these are the columns you got in the result section. Properties, scale, and about substitutions. So is it clear for everyone? Or if you want to do this exercise, I'll give you the sequences. If you need it, I'll share the sequences here. So I'll share for both protein and nucleotide. You can try these things later on. So these two sequences are DNA sequences.
Ma'am, can you explain about that physiochemical properties, conformational properties, and nucleotide content? What all these are?
Okay, so sir, are you going to do that?
No, actually.
No, I will. Okay.
Mehta, you are done almost?
Yes, sir, it's done. Almost done.
Okay. Maybe we'll wind up now. Yeah.
Yeah, so, okay. So I'm sharing you the information in for like test cases. If these are like I'm sharing 2 sequences for proteins. If anyone wants to try that later on, they can do that. So try to write those in faster format, then only the tool will work and you can get the property values. So I think I'm done from my end. Sir will continue. Hello.
Hello.
Tell me.
Yeah.
Okay, so I think we have done sufficient lot of exercises today, and hope you enjoy the session. And if you have any questions, just you ask me anytime, or you can practice again at home. And we will see you tomorrow. Tomorrow I will continue the session. Okay.
Yeah, sir. Just a minute. Just before we let you go. Yeah, just a minute. Yeah, I would thank once again our expert Professor Gromiha sir and his team members. I would like the team members to introduce once again. I would really express my thanks on behalf of our director, institute, and participants for the team members. Yeah, kindly introduce yourselves. We are really thankful for you.
So I am Medha. I am PhD third year. I am working under Professor Grumia, and I am working on machine learning. And if anyone has any doubt later on, they can contact us. They can check our lab website and they can contact us through mail. Yeah, thanks.
Yeah, yeah, thank you. The other member, other scholar.
Yes, sir. So I am Kulandya Sami.
Yeah.
I am doing PhD here. So I am working on membrane protein mutations. So we have developed several databases and prediction algorithms in order to account the effect of mutations in the membrane protein structures, stability, as well as the diseases.
Yeah. Okay. Medha madam and Kulandya Sami, thank you for your sessions through this lab, and I would request participants to appreciate their efforts. And one more highlight which I would like to point out is that this workshop is highly interdisciplinary. There may be experts from various fields like mechanical engineer, mathematics. Then there will be the other extreme, biology, chemistry. Then there will be that computer science. There will be that information technology. Okay, so this being a very highly interdisciplinary, and that is why I think this has been sponsored, and government is more, they want to polish the talent of the faculty members. So there may be some lot of gaps between one extreme and the other extreme. So instead of taking things to the extreme, we must take this positively, and we have to put our hearts to understand the concepts. The mathematics persons must try to understand the biology. The biological person must try to understand the mathematics. So that's how this interdisciplinary subject works. So we have to apply ourselves. Once again, I ask all participants give a very applause to this efforts by this research scholars and their mentor. Yeah, thank you once again for all of you. If participants wants to thank, they can thank. You can unmute and you can just thank this research scholars. It will be a great deal in boosting their morale. It's not so easy to convince everybody, because being highly interdisciplinary, it's very, very difficult. Yeah. Please, participants, you can put in your applause, please.
Thank you, Medha ma'am and Sami sir. It was very nice. You explained very well. And yeah, since I'm from chemistry background, so it was a little bit difficulty, but you explained us very well. Thank you so much.
Hello.
Yeah.
Thank you to the entire organization. Could get guideline or something so that we can have the hands-on practice done properly. Actually, we are from backgrounds of study, so it would be really helpful for us if we could get a manual to the hands-on session that we had today. Thank you.
Thank you, Medha ma'am, and thank you, Kulandai sir. Big round of applause for the efforts you have taken to explain us.
Yes, sir. Thank you. Thank you once again. So we will join for tomorrow's session right there at 9 o'clock. So I request all to join by say 8:55 or so. Yeah. Thank you. Thank you, everybody. Yeah, I stop the recording here.
Bharat sir, excuse me, sir.